Okay, so today we're going to cover, I'm going to guess like one more topic before the exam. I don't expect that we'll get to the very end of this muscle packet. So if you want to pull up the practice exam, I think there are probably about 3 questions that we'll probably kick out of that. Good. But as we get rolling, a reminder of where we were last time. So we were talking about how muscles are structured, right? So we know already that their skeletal muscle is the target of our somatic branch of the nervous system. So one of our 2 pieces of the motor part, the effector part of the nervous system, somatic versus autonomic. Somatic goes to skeletal muscle. Skeletal muscle's job is to move bones towards each other to make your body move. It does that by contracting sarcomeres. Our sarcomeres are made primarily up of 2 types of filaments, so proteins. We have actin making up a thin filament. Here's the structure of actin. We have our myosin making up a thick filament linked together in the middle of the sarcomere by the M line. It looks like this. It's got this structure called a cross bridge on it with an actin binding site and a place for ATP to come in. I'm going to flip back for a second. On the thin filament, we've got a binding site for myosin that during relaxation, right, so when you're relaxed, is covered over by this tropomyosin molecule. That's this one skinny one. We've got this troponin complex right next to it. It's basically our switch for moving the tropomyosin and exposing those binding sites. Okay. So here is that structure of the sarcomere. So the sarcomere works by sliding these proteins past each other. Right? So neither of the actual proteins, myosin or the actin, change in size. Right? So here's my pencil, it's the myosin. My mouth is about to be the M line. My fingers are the actin. We're doing this. Right? So the pencil didn't change in size, so the myosin doesn't change in size. My fingers don't change in size, so the actin, that thin filament, doesn't change in size. What changes is how much we're overlapping these filaments. Okay. We named different pieces of the sarcomere based on what is and isn't overlapping in these regions. Okay. Any questions about the structure of the sarcomere or the sliding filament model before we move on to the next topic? So hopefully you reviewed this after last time. Okay. Cool. Feel good about that. Next, we're going to be talking about the cross-bridge cycle. Okay. So this is how we actually do that, right? Like this is how we actually slide these filaments. Right? So the cross bridges are the name for that paddle, that little golf club on the myosin. And we're going to be using them. Pick a different one because I told you that that pencil is myosin. We're going to be using them to take our actin, which is the Z line, so this green pen is now what my fingers were before. Okay, so we're going to take this and we're going to flick it towards the center. That's all we're doing in the cross-bridge cycle. That is our goal. Okay, that's how we make our sarcomere shorter. Okay. So we're going to have a series of steps where we get actin and myosin binding to each other. And then we're going to have our myosin change shape. So just like I had to bend my wrist to throw that pen, same thing's going to happen with the myosin when we try to throw the actin towards the center of the sarcomere. Okay, and that's how we contract a muscle. Okay. So you can also think about this as rowing a boat through the water. Our cross bridge is our paddle or our oar, right? And we're going to make a step of, our cross bridge is like this. We're going to make contact with the water, and then we're going to push through the water, moving things together. Okay. So we're going to have a paddle. We're going to have to link our things together. So we're going to have to grab hold to the thin filament, to the actin. Right. Then we're going to have a power stroke. So we're going to have actual movement of the myosin, make this propelling motion. And then eventually we're going to have to let go. Right, because we do want to reset this, and actually to make the sarcomere really shorten, we're going to have to do this a bunch of times. right? So we don't just flick once and then throw the pen all the way across the room. It's like, we got a lot of cross bridges and they have to break and form and break and form. To do this slowly, right? You want your muscle to be able to move slowly. So we got a cycle. I want you to take a picture of the cycle in your notes and try and make some sense of it on your own before I walk you through the steps. So I want you to look for that first step. I want you to look for what is bound to the myosin at that point in time where we have our first step of the cross-bridge cycle. There might be a couple things. So check everything that is bound to the myosin that you can see in the image for that first step. Ken, or you? Anybody see right now? I didn't hit anybody, did I?
No.
Okay. I might go some more things. Hopefully I won't hit anyone. All right, take another second and we'll walk through it. Okay, cool. Yeah, so in the first step, when we start this cross-bridge cycle, we're going to be already bound to actin. We're going to see that we have ADP and a phosphate ion bound to the myosin as well. So we're going to look at the picture. Okay. So first thing to notice about this picture is that number one is not at the top, right? So number one, we're starting down here. So our first step is when we bind our myosin to the actin. So we can see here the setup. I'm going to point out to you, right? So when we're getting ready to do this, we've uncovered these myosin binding sites on the actin. So they're currently exposed. They're not covered by the tropomyosin. And you'll notice that our troponin molecule looks a little different. It's got this green hat. That green hat is calcium. I'm just pointing these out because that'll come back in the excitation contraction coupling part of this slide set. Okay, so we first are binding our myosin with its actin binding site onto the actin at that point where we have our myosin binding site on the actin. Okay. At this point in time, we have ADP in that ATPase site, as well as a phosphate ion. Okay, and so we're remembering that when we're looking at ATP and ADP, we've got a molecule, there's the adenosine part, and we've got two phosphate groups for ADP. The D is for di, so this would be ADP. And if we want to make ATP, we add another phosphate group. Okay, so that in full up there would be ATP. Okay. So what this means when we're looking at this image and we can see ADP and a phosphate ion here is that we've already broken down an ATP molecule and gotten the energy from this bond. Okay, cool. So we're starting out. I'm not going to use this ore model because I like throwing stuff. Okay, so here's my arm, here's my myosin. I'm holding on to my actin, which is my myosin. Right, so it's out like this in our high energy form, already ready to throw for our first step. Okay, so that's what we got here. Okay, so that's what we saw in this first step. We bound the myosin to the actin. So that first step was when we grabbed on our eraser, grabbed onto it. All right. So the next thing that happens is that our phosphate ion falls off. Okay. Imagine we're actually like this, right? So we're going to have a piece of the stuff bound so that my myosin has fall off, right? So my hair type. So this is going to change shape, right? Whenever we have something bind or unbind to a protein, it changes its shape. Which is what leads us into this next step. So this is the power stroke, right? So when that phosphate ion fell off, this changes the shape of that cross bridge, right? So our cross bridge was out here in the higher energy form. When the phosphate ion falls off, that's our power stroke. So that's literally when we get that snap. That's what just happened. So when we see this power stroke, right, we move our actin towards the center of the sarcomere because now we've changed this shape here, right? You can see that now we have a bend, right? That cross bridge is bent in. And we don't have a phosphate ion bound here anymore, which is why we have that shape change. Cool. All right, so after we do that, we're done with having stuff bound in right now to our cross bridge. So we let go of the ADP. Right, so we're emptying out this site, this ATPase site, because we're going to get ready to grab more energy so that we can eventually do this again. But first, we can see that when we get rid of that ADP, we go into kind of like a low energy form. So when we're in this state where we don't have ADP bound, we don't have a phosphate ion bound, and we're already in this bent shape, right? Like I'm stuck like this right now. I can't like bend more, right? I don't have any energy to pull that actin further towards the center. And I'm not even in the position that's going to give me force, right? So this is our low energy form of the myosin, and this is called rigor. So that's our third step. So when we're kind of like stuck in this place, we've already pushed the actin towards the middle, and we've let go of the ADP and the phosphate ion totally. Now we get to bind new ATP. Okay. So new ATP is coming in, and it's going to that site we emptied out. Right, so we emptied that site out. Now we bind on ATP to that ATPase site. Right, so this is at that same corner where in the beginning we had our phosphate ion and ADP. Right, so that's that same spot. Right, and because we bound something in, we have a shape change. Right, so when we put the ATP molecule there, that disconnects us from our active. So we were stuck like this, and we didn't really like throw and fully let go like I did just there. Right, we were like this in rigor. When the ATP binds, we let go. We're getting ready to grab it. Right, so we're like detaching in step 4. Okay, so now we hydrolyze the ATP. Right, so ATP hydrolysis, anytime you see this lyse ending to something, okay, anytime you see that lysis, that lyse ending, that means we're breaking. So ATP, adenosine triphosphate was this. That's what we have bound in here at the corner at this ATPase site right now. Hydrolyze it, so we break it down using water. Now we've got ADP again. Okay, so when we get that breakdown of ATP to ADP and phosphate ion, we're going to tuck in that myosin head. So this is the tucking of the myosin head formally, putting it into this high energy form. Right, so we're all like reached in like this. I don't know what position to put my arm in exactly here, but we're like ready to grab onto the actin again and throw it again. Right. So here in this high energy state at the end, right before we start step 1, we're ready to grab onto actin again, but we haven't done it yet. We've got that energy ready in the myosin. We're in this high energy form. all we have to do is grab actin, drop the phosphate ion. So we do this power stroke, throwing it towards the center again, and repeat and repeat and repeat until our sarcomere has shortened as much as we want. Questions about this process? Like, it looks complicated and sciencey and has hard words in it when you look at it in the image. But if you think about it in terms of like physical motion, it's kind of what you would have to do. I want to make sure that it is. Good. Okay. So looking back at that, what caused the myosin to pull the actin towards the middle of the sarcomere? All right, take another second and I'll take a look. Okay, great. Yeah. So we started out with our high energy form of myosin, ADP and a phosphate ion bound in on the ATP space site in our first step, right? As we bound to the actin, phosphate ion fell off, leading us into the second step, the power stroke. Then we have the ADP fall off, right? So we're emptying out that site. We're stuck for a second in this uncomfortable low energy position called rigor until we bind in a new ATP molecule. We get to let go of the actin. We get to break down the ATP, putting us into high energy form, ready to go again. So that's the real point of the cross-bridge cycle, and that's really all it is. So you want to learn the steps, you want to know what happens and why, but that's all we're doing. So now linking this up to our nerve. We talked about the neuromuscular junction, what's going to here. And we talked about the cross-bridge cycle, so we talked about how we use the energy to shorten the sarcomere. We want to link those two together. So that's what we mean by excitation contraction coupling. We're thinking about, okay, so we've got our acetylcholine coming in here. That's our neurotransmitter from the somatic nerves responsible for exciting our muscle cells. So we have our end plate potentials. They're always depolarizing. They're always reaching us toward an action potential, right? Because that's the whole point of a nerve to a muscle. Now we want to think about how do we get from this action potential that we started by releasing acetylcholine, depolarizing our muscle cells, to actually contracting and doing this cross-bridge cycle. So basically, we're going to be coming across the membrane with this action potential, this depolarization. A wave just like with our action potentials in neurons. But now we're going to have that action potential triggering events tunneling into our muscle fibers, into our muscle cells, causing the release of calcium, which exposes our myosin binding sites on actin, at which point we can bind together the actin and the myosin and do the whole rest of it. So this is going to be dependent on having a nerve tell your muscle to contract. So this is depending on our neural input from our motor neurons, which are somatic. We're going to need calcium specifically for this. Okay. So the steps. Big U. We already signal at the neuromuscular junction. So our motor neuron release acetylcholine. acetylcholine from its axon terminal, sometimes called the terminal baton, right? Released that acetylcholine. We caught it with a nicotinic cholinergic receptor, created a depolarization, created an action potential. Okay. So we've got an action potential at that motor end plate, so that little sort of squiggly pocket where we had the nerve come in. Now we're going to send that action potential down the sarcolemma. The sarcolemma was just a fancy name for our plasma membrane on our muscle cell. Okay. So we have our action potential going down the membrane of our muscle cell. Then it's going to go down a T-tubule. So we have those tunnels, they were green in the image of them, those tunnels, those T-tubules tunneling into our muscle cell. Action potential travels down. So it's going across the membrane, and then it's diving down a tube into our cell. We're then going to have receptors in those tubes, and those receptors are going to open calcium channels. And so we're going to release calcium a couple places, but our main one that we're interested in is we've got calcium stored in our sarcoplasmic reticulum. There's a modified version of our endoplasmic reticulum, but we're really thinking about it as like bags of calcium on either side of the T-tubule. Okay. When we release this calcium went somewhere, it went into the cytosol, so it's swimming around in your muscle cell now. When you have calcium swimming around in your muscle cell, that means it's going to touch stuff. One of the things it touches is tryptonium, that little snowman guy. So it's going to put on his calcium hat. Okay. That means it's going to act as a switch, push our tropomyosin off the myosin binding site on the axon, exposing those sites so that we can start cross-bridge cycling and start flicking that axon towards the center of the structure. So look at the pictures. Okay. So reminder of the pieces that we did before, because it was a lot of words, and I'm not sure I was most on it the day we talked about that part. Okay, so we had the action potential coming down a motor neuron. So this was our somatic nerve. Okay, so that was our action potential. Just like our synapses in the central nervous system were, but now we're connecting to a muscle. Okay. So we had an action potential. When we have voltage, the action potential in this axon terminal, that means we open a voltage-gated calcium channel here on the axon terminal. Right? So calcium comes in. Okay. That's our trigger for releasing our acetylcholine. All right, so here's our acetylcholine in a synaptic vesicle. Then we have the exocytosis, so letting that acetylcholine leave our first cell, our motor neuron, end up in this little cleft here, floating around, where it can bind to our acetylcholine receptors, which happen to be nicotinic cholinergic receptors here on the motor end plate. When we talked about this before, we talked about the fact that those nicotinic cholinergic receptors act as channels, basically. So they open when we bind the acetylcholine. Different color for acetylcholine. All right, so we open a channel. So primarily the action that happens then is sodium comes in. We also have some leakage of potassium out, but that doesn't really matter because potassium moves slow, sodium moves fast. Lots of sodium moves. So that caused our end plate potential, which was basically a graded potential, so like a little potential, not a full action potential, but a bit of depolarization. So that was the EPP in here. But eventually that builds up and we get an action potential on the sarcolemma. Okay. Question about that process that we did before we get to this part. Hopefully you can walk through the steps. We have them listed out in the thematic packet. But that's how we got to where we are. So we've got a little bit of that repeating here on the steps of our excitation contraction coupling, because we're coupling, we're connecting our excitation, which is the nerve emitting neurotransmitter, to the contraction, the cross-bridge cycling. You can see that cross-bridge cycle happening here in the middle. So this is our linking between the nerve stuff and then what's happening inside the cell. Okay. So we start here in these steps with that release of acetylcholine. So our somatic nerves are always releasing acetylcholine. Remember, we saw acetylcholine from other places. We saw it always between any preganglionic neuron and any postganglionic neuron, whether we were talking sympathetic or parasympathetic. We also saw it coming from our postganglionic neurons of the parasympathetic nervous system going to the effector organs. We had a different type of receptor on the effector organs at the end of the postganglionic parasympathetic neurons. That was the one place we had muscarinic cholinergic receptors instead of nicotinic cholinergic receptors. So unless it's that one spot, it's the nicotinic kind. This opens an ion channel. That's at the end of the parasympathetic pathway. It's muscarinic, they're deprotein coupled, and we're so we got acetylcholine. We got a nicotinic cholinergic receptor here on our motor end plate. So we start an EPP, an end plate potential, right? So this is our end plate. Potential is telling us, triggering our memory, that this is a change in voltage, right? So depolarization. So this kicks off an action potential. Okay, so here we have our action potential. So this is a wave of voltage, basically, going down our sarcolemma, away from that motor end plate site, just like kind of all across this motor fiber. And it's going down our T-tubule. So our T-tubule is a tunnel into the cell. We're coming down. And you can see how that works, right? Because we just had that membrane kind of continue into the T-tubule, right? So hopefully, not a big deal, right? Like we just followed this road, we just followed the membrane into the T-tubule. So we've got our action potential going down into the cell. Right. So when we looked at the overall structure of a muscle, we named this as a triad. So a triad was the T-tubule in the middle with the lateral sacs of the sarcoplasmic reticulum on either side. I'm drawing this as a big T because this is the T-tubule. On either side, we have sarcoplasmic reticulum. That's why I made the top of the T so big. This is our triad. Okay. So I've got this T-tubule in the center going between these lateral sacs of the sarcoplasmic reticulum. We can see they touch each other, right? Which means that when we have an action potential come down the T-tubule, we can do stuff here to our sarcoplasmic reticulum. So step 3 is we do stuff. Right. We're not going to worry about the actual receptor type, although it's listed in those number steps on the previous slide. What I want you to know is that when we had this action potential come down, we had a bunch of calcium stored here in these lateral sacs. Okay. And when we have the action potential, that's our trigger to release it. Okay. So we released a bunch of calcium. It's now swimming around inside of our muscle cell, swimming inside the cytosol here. Okay. So swimming inside the cytosol, which means stuff touches it. So stuff binds to it. Okay. So here we see our thin filament, our actin, right? That's our thin filament. Okay. Hint I thought of the other day when we were thinking about sarcomeres, right, is that our actin, right, if you were to label like A to B, A comes first, so actin comes first at the Z line, and then our myosin at the M line is in the middle. That may or may not help you remember those structures on our sarcomere, but I'm pointing those out so that you can orient yourself to this thin actin, right? So that was our thin filaments at the ends. Okay, that's what we're what we're looking at. Okay, so when we bind our calcium here, our original state, our relaxed state of actin, we would have had our G actin linked into F actin, like beads. We have a spot for myosin binding on it. So this is our myosin binding site on the actin. Okay. Right. I'm just redrawing the picture, but telling you the different pieces one by one. Right, so that was the myosin binding site. But when we were relaxed, we have our tropomyosin covering over, so they're blocked. Right, so that was the tropomyosin. Okay. Pick a different color for our troponin to make it clearer. It'll be red. Okay. So our troponin was sitting here next to the tropomyosin. It had three parts. Don't worry about them. It's holding onto the troponin. It's holding on to the troponin is holding on to both the tropomyosin and the actin. It's the important piece. Okay. So it's sitting here. That's our troponin. Okay. So this was what was happening at rest. You can see there's no calcium in this picture, right? What changed here as we move into this state where we're ready to cross-bridge cycle is that when we add calcium to the cytosol, it's binding here on top of the troponin, right? So we add calcium here, right? So we add a calcium, and this shifts the tropomyosin. So hopefully this will erase my whole tropomyosin line. So as soon as we have calcium here, our tropomyosin gets pushed over. Oh, that's an eraser. So now that we have the calcium, we're going to expose those sites, right? Because we want to bind to myosin. Calcium is our trigger that it's time to contract the muscle. So now we expose those myosin binding sites, right? So I have redrawn this picture that you have down here in step 4. You can see those exposed binding sites in addition to the calcium bound to the troponin. Tropomyosin is still there, but it's not covering over our binding sites. Okay. Which means we can grab it. So we grab it with our cross-bridge cycle, right? So here you can see the cross-bridge cycle starting. This one's our myosin, right? So we had our myosin binding sites on the actin. And we had our actin binding site on the myosin, right? So that's at that point of contact. And they're just like magnets, they go together. Okay. So here we can even see an image of a cross bridge that's in the high energy form, ready to grab the actin, right? But we can see that on several of our other cross bridges, we've done that first step of the cross bridge cycle at least, right? So we grabbed onto the myosin, and I suspect we have also thrown it forward in the power stroke, right? So change the shape, right? And then we're getting ready to reset these, right? Bind in ATP, let go, break it down, cock ourselves back into this high energy state and do it over and over again. So this image isn't showing you necessarily any particular step. It's showing you that the cross bridges are forming and reforming. So each of those little hands is helping out, doing its own cross bridge cycling when we're able to, because we have this exposure of the myosin binding site. Okay. So we do this over and over and over again. As long as there's calcium in your muscle fiber, you're going to keep doing this because this just depends on the fact that we have those sites exposed and we're breaking down energy, right? But the only control part was yes, calcium or no calcium. Okay. So in order to stop this contraction, we're going to have to get rid of the calcium. So the final part here is that we are going to actively transport our calcium back into our storage location, so back into the sarcoplasmic reticulum. We're using energy for this, so this is active transport. Right, we're sucking up the calcium and putting it back in the sarcoplasmic reticulum, which will allow our cross-bridge cycling stop, slow down, so that you can stop shortening the sarcomere and get ready to do this again, ready to relax, ready to use your muscles however you want. Okay. So when the calcium is gone, we don't have calcium sitting on that troponin, we go back to our original state, right? So there's no calcium here on the troponin, which means that our tropomyosin is covering those myosin binding sites. No cross-bridge cycling possible because the spot for that connection is now blocked. Okay, so questions about this? So this is a zoomed in picture of what the actin, tropomyosin, and troponin complex looks like when you're in a relaxed state versus a contracting state. So this is what I was drawing up in the top corner, right? So we have, sorry, this is covering it around the screen. Drag this out of the way, right? So our tropomyosin is our long string. Our troponin is this switch, this little stack here. It's holding on to actin, it's holding on to tropomyosin, and it's got a spot where we could bind calcium. Okay. When it's relaxed, there's no calcium. As soon as we have calcium, we bind the calcium to the troponin, which shifts it over a little bit, right? It changes shape. Everything changes shape when you bind something to it. This particular shape change means that it pushes the tropomyosin out of the way, off of those binding sites, which means the cross-bridge cycle can occur. Okay. So in order to stop contracting, we have to get rid of our calcium, right? So it has to leave the troponin. Means when it leaves the troponin, we cover back those myosin binding sites on actin. So we're blocking that action, we're blocking that connection. And we got to get rid of all the calcium that's spinning around our cytosol because that was what was blocking, or that was what was causing this changed position of troponin. So we have something that does that, like a pump, right? Active, actual movement of that calcium back into the sarcoplasmic reticulum, which is where we're starting. Let's take a look at this in action.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as the sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
That is enough new topics for you to be studying for the exam, certainly. So we're going to leave the twitch and the different types of contraction for later. So I'm going to pull up the practice exam and tell you which questions those corresponded to, so you don't worry about them. I'll remove them and replace them after class, but I'm just going to let you take a look real quick. Okay. I'm pretty sure I tried to put them at the end for you. Okay. Okay. So we're going to kick out this question about the isotonic contractions. We'll do isometric and isotonic contractions next time. This is also a question about twitches and contractions. So question 34, where it asks you about tension again. Basically, if you see the word tension, we're going to kick out the question. So we'll kick out 34 here with the tension, and we'll kick out this last one about motor units. So if you've already done it, those are the ones that we're not going to cover. For those of you that do it this weekend, I'll get them off of here so they don't confuse you and try to swap them. Okay. We do have 5 minutes left. So is there anything people want to go over one last time before we get that? So I will list major topics. How about that? So let's pull up the whiteboard. Okay. Ooh, obviously. What? Okay. No. All right. I'm going to bring up MS Paint and just share my screen. All right, one more time. Okay, there it goes. Okay. So we started by talking about branches of the nervous system, right? And the overall structure. So you want to be able to tell that we have a central nervous system, that was brain and spinal cord. We have peripheral nervous system. That's all the branches we've been talking about. We have afferent nerves going in. We have efferent nerves going out. And this is where we have our next split. So in our efferent nerves, we're going to split between somatic nerves and our autonomic nerves, ANS. Okay. The way we tell the difference is where we're going primarily. So skeletal muscle is somatic. So this is under voluntary control. Autonomic nervous system is smooth muscle. Your cardiac muscle, so your heart, and glands, possibly fat. Okay. So this is all involuntary. This is all homeostasis. We had two pieces of this autonomic nervous system. We have our rest and digest part. That's our parasympathetic nervous system. And we had our sympathetic nervous system, which is your fight or flight, your kind of stress part. We have dual innervation. So both of these are going to these same places. And we are just switching which of them predominates. So which of them is more in control, not totally turning on and turning off. So we have this trade-off. We have more stressful situations, sympathetic, more relaxing situations, parasympathetic. Okay. We talked about a lot of differences between them. One of the main ones was general structure, so where they come from. Sympathetics were thoracolumbar, parasympathetics were cranial nerves and some sacral nerves, so craniosacral. So parasympathetics from the top and bottom, sympathetic nerves from the middle. We talked about pathways. So sympathetic nerves generally have a sympathetic chain that lies parallel to your spinal cord. So we have a short preganglionic nerve to the sympathetic chain, then long ones out to all the places they go. We can also have collateral ganglia. A ganglion is just somewhere you have cell bodies, right? So that would just be a ganglion somewhere closer to an organ. You'd still have a preganglionic or postganglionic neuron. Sympathetic nerves also go to the adrenal medulla. They have a preganglionic neuron going to the adrenal gland all the way inside. We have a modified cell called a chromatin cell inside the adrenal medulla associated with the sympathetic nervous system. Parasympathetic nerves, long preganglionic neurons, basically to the organ itself, then a very short postganglionic neuron into the organ. So, I wasn't writing that because next is neurotransmitters. Right? You want to know the roots, but this can go on this map already. So imagine each of these arrows. First one is a preganglionic neuron. Second one is a postganglionic neuron, right? Because that's our general structure. Okay, so both of these in this first step between pre and postganglionic neurons release acetylcholine. Okay. Which means both of our postganglionic neurons have cholinergic receptors on them, right? Which also has the CH in it, cholinergic receptors. They're both nicotinic. Same thing for the somatic. So acetylcholine and our cholinergic receptors. For our parasympathetics, acetylcholine is also at the endpoint for our postganglionic neuron. So here we have acetylcholine. Okay. Different type of receptor. Still a cholinergic receptor, will be a muscarinic one, so with an M, as you get to the smooth muscle, cardiac muscle, etc. Sympathetic nervous system, more complicated, because we have a new neurotransmitter. at the end here, right? So this is where you see primarily norepinephrine. So that's for the normal routes through the sympathetic chain or the collateral ganglia. If we're going to the chromaffin cells in the adrenal medulla, we'd have epinephrine, which in some places is called adrenaline, coming from the adrenal gland, right? So that's how you can remember that piece. I'm going to draw a triangle because that's the shape of the adrenal gland. So that's where epinephrine comes from. Okay. We are at time, so you guys can leave, but I can also keep talking a little bit. So I'm just going to let you know that another major topic you're going to want to do after this is receptor types. We have a bunch of adrenergic receptors, the alpha and beta types. Then we went into endocrine. We talked through all the glands. You really want to focus on hypothalamus pituitary stuff. You want to be able to understand how the hypothalamus in particular signals to the anterior pituitary and those two pathways to the thyroid gland and the adrenal cortex were the ones we really zoomed in on. So focus on those. And then we moved into somatic stuff and muscle stuff. I was just reviewing it while we were talking about this. And this.